Hello, my name is Professor Hudson, and you are about to view narrated PowerPoint lectures for Chapter 12, which covers DNA, the molecule of heredity, and deals with structure and function of DNA. We will also talk about Chapter 13, which deals with the central dogma or central concept of genetics on how the information in DNA is used to make protein. For Chapter 12, the following sections will be covered in this narrated PowerPoint lecture. 12.1, how did scientists discover that genes are made of DNA? 12.2, what is the structure of DNA? 12.3, how does DNA encode genetic information? And 12.4, how does DNA replication ensure genetic constancy during cell division? This narrated PowerPoint lecture will also cover information found on pages 216, 217, and 218 of your textbook. Please make sure you read those pages. They're not on the syllabus, but they are covered with this lecture, and they cover structure and function of DNA. Let us begin. Let's first take a look and talk about some history regarding how DNA and its organization was understood. By the late 1800s, scientists had a limited knowledge of genes. Genes are heritable information packed in discrete units. Genes determine many inherited traits. Scientists were unable to determine the gene itself. By the 1900s, scientists learned that genes are part of a structure called chromosome. And the chromosomes are made up of deoxyribonucleic acid and protein. So little by little, scientists were beginning to understand the organization of genetic material. People carry chromosomes in the nucleus of their cells, and this chromosome is made up of deoxyribonucleic acid and protein. And within the chromosome are chunks of information that encode for physical traits. And these discrete chunks of information are called genes. Let us review. Question 1. Which list is ordered from largest to smallest in size? Choose the correct answer and make sure you write down the letter that corresponds to this correct answer, as you will need it for the lecture quiz. Transformed bacteria revealed the link between genes and DNA. Prior to this point, people knew what DNA was, but they didn't really understand what it was about it that allowed it to work the way that it did. In the 1920s, Frederick Griffith worked with two strains of Streptococcus pneumoniae bacteria. The S strain causes pneumonia when injected into mice. The R strain does not cause pneumonia when injected into mice. When the S strain is killed and injected into mice, it does not cause disease. Griffith made a sample of the heat-killed S strain and mixed it with the R strain. He injected the combination into mice and noticed that the mice got pneumonia and died. This was unexpected because the heat-killed S strain by themselves normally are harmless, and the live R strain are normally harmless by themselves. But for some reason, when the two were mixed together, the mice got sick. The results of Griffith's experiments led to several new deductions about the genetic material. Some substance in heat-killed S strain bacteria changed the living harmless R strain bacteria into the deadly S strain. So something about the S strain bacteria, even after they had been heat killed, appeared to have changed the harmless R strain bacteria to make them deadly. This substance that caused this transformation might be the long sought material of heredity. In the 1940s, Oswald Avery, Colin MacLeod, and Macklin McCarty isolated DNA from S strain bacteria and mixed it with the live R strain bacteria and produced the live S strain bacteria. This suggested that the transforming molecule from the S strain was DNA. To rule out the possibility that a protein contaminant was actually causing the transformation, Avery treated samples with protein-destroying enzymes and still induced transformation. So, just in order to make sure it was really nucleic acid and not a protein that caused the transformation in the bacteria, they treated the sample with an enzyme that destroys protein. Yet, this material was still able to transform the bacteria, so clearly the material was not a protein. However, when they added enzymes that destroy DNA, then the transformation did not occur. So this suggested that whatever this transforming material is that was passed from one strain of bacteria to the other, it had to have been a nucleic acid. Griffith's experiments can be explained if DNA is the transforming agent. Heating the S strain cells killed them but did not completely destroy their DNA. When killed, S strain bacteria were mixed with living R strain bacteria. Fragments of DNA from the dead S strain cells became incorporated into the chromosome of the R strain bacteria. If these fragments of DNA contain the genes needed to cause disease, an R strain cell would be transformed into an S strain cell. Thus, Avery, MacLeod, and McCarty concluded that genes are made of DNA. This slide illustrates the process that might have been going on during the experiments done by Avery, MacLeod, McCarty, and Griffith. In this figure, you see a bacteria with its single chromosome. If, for example, the heat-killed S strain released some DNA fragments that were able to be taken up by the bacteria and incorporated into its chromosome, then whatever traits were on those fragments from the S strain bacteria will now become part of the genetic makeup of the R strain bacteria. This is called transformation. This process is also used in labs every day in order to get bacteria to express genes that they normally would not express. Question 2. If a bacterium causes pneumonia, what would you expect of its offspring? Choose the correct answer. Let's now take a look at the structure of DNA. Knowing that DNA is made up of genes does not provide an answer to the critical questions about inheritance. The secrets of DNA function and therefore of heredity itself are found in the three-dimensional structure of the DNA molecule. DNA is composed of 4 nucleotides. DNA is made up of chains of small units called nucleotides. Each nucleotide has 3 components. Those components would include a phosphate group, a sugar, and in the case of DNA, that sugar would be deoxyribose, and one of 4 nitrogen-containing bases. So this is the basic structure of a nucleotide. There can be 4 different bases for DNA. You have your adenine, guanine, thymine, and cytosine. They're sometimes also pronounced adenine, guanine, thymine, and cytosine. These four bases make up the language of DNA. In the 1940s, Erwin Schargraf, a biochemist at Columbia University, analyzed the amounts of the four bases in DNA from diverse organisms. He discovered a consistency in the equal amounts of adenine and thymine and equal amounts of guanine and cytosine for a given species, although there was a difference in the proportion of the bases. This finding was called Schargraf's rule. Bases that bond with each other are called complementary base pairs. So basically, he determined that if there's an A or adenine on one strand, there must be a thymine or thymine on the other. And if one strand has a cytosine, then it must be a guanine on the other. So basically, A will pair with T and G will pair with C. Those are the only base pairings allowed in order to maintain the structure of the DNA molecule. Because of their structures and the way they face each other, A can only pair with T and G can only pair with C. Complementary base pairing explains that for a given molecule of DNA, the number of A's must equal T and the number of G's must equal C. Another way of saying this is that no matter how many of the guanine bases are present, for whatever number they're present, it must be the same amount of the cytosine bases. DNA is a double helix of two nucleotide strands. In the 1940s, several other scientists investigated the structure of DNA. Rosalind Franklin and Maurice Wilkins studied the structure of DNA crystals using X-ray crystallography and X-ray diffraction. They bombarded crystals of purified DNA with X-rays and recorded how the X-rays bounced off the DNA molecule. The resulting pattern does not provide a direct picture of the DNA structure, but the researchers were able to extract specific information. Wilkins and Franklin deduced the following about DNA from these X-ray patterns: that the DNA is long and thin with a uniform diameter of 2 nanometers. It is helical and twisted like a corkscrew or spiral staircase. They figured out it's a double helix and that it has repeating units. Phosphates are probably on the outside, they figured out. However, it was James Watson and Francis Crick that actually combined the X-ray data with bonding theory to deduce the final structure of DNA. Watson and Crick proposed that DNA is a single strand of polymers called nucleotides, and that you have two of these strands wrapped around each other in the form of a helix. Within each strand, the phosphate group of one nucleotide bonds to the sugar of the next nucleotide in the same strand. He figured out that the sides of this molecule are made up of alternating phosphates and sugars, and that the center of this molecule consists of bases that have hydrogen bonded together. All the nucleotides within a single strand are oriented in the same direction and thus have an unbonded sugar at one end and an unbonded phosphate at the other end. The two strands of nucleotides that are wrapped around each other run anti-parallel or in opposite directions.